Hi, my name is Francesco Ricci. I'm a postdoc researcher at the Bioelectrochemical lab of Professor Palleschi at the University of Rome Tor Vergata. Today, together with my colleagues, I'm going to show you preparation of an electrochemical immunoassay for the detection of trichothecenes mycotoxin in food samples. Among the different methods used in immunoassay tests, enzyme immunoassay is one of the most popular. In this method, either the antibody or antigen is labeled with an enzyme. Recently, the use of electrochemical methods for the detection of the enzyme product has demonstrated to be particularly advantageous for certain applications. Electrochemistry is in fact less prone to interferences, is suitable for miniaturization and for mass adduction at low cost. In this perspective, the use of screen-printed electrodes is of great importance to ensure low cost and easiness of procedure. Moreover, the use of magnetic beads could be coupled with screen-printed electrodes in order to obtain the best sensitivity of the method. Today we will show you how to prepare an electrochemical immunoassay based on the use of screen-printed electrodes and magnetic beads for the detection of trichothecenes mycotoxin in food samples. Trichothecenes mycotoxins are commonly found in cereals and for this reason represent a serious threat for the safety of cereal-based food and feedstuff. The most important trichothecenes within group A are T2 and HT2 toxins. As we will see, this method can be easily performed by non-trained personnel and with low cost and portable instrumentation. This is one of the main objectives of the integrated project BioCap,